Welcome. In the quiet hours, when the day's noise fades, some of the most intricate work in medicine unfolds at a scale almost beyond imagination. Particles smaller than a single cell, engineered with care and precision, navigate through a patient's body, carrying treatments to places once unreachable. Please join the Z-Files as we learn more about the story of nanoparticle therapy, one of patient science, clever design, and a gradual reset of medicine's boundaries. Part 1. The Scale of Possibility. To understand nanoparticle therapy, we begin with size. A nanoparticle measures between 1 and 100 nanometers, a range so small that 1,000 of them could sit side by side across the width of a human hair. At this scale, materials behave differently than they do in larger forms. Surface area dominates. A gram of nanoparticles presents vastly more surface to the surrounding environment than a gram of the same material in bulk, and this changes how they interact with cells, proteins, and the body's defenses. Nanoparticles used in medicine are crafted from a handful of material families. Lipid-based particles resemble the fatty membranes that surround our own cells, which makes them gentle and often well-tolerated. Polymeric nanoparticles are built from long chains of repeating molecules, offering flexibility in design and the ability to degrade predictably over time. Metallic nanoparticles, often gold or iron oxide, bring unique properties like heat generation or magnetic responsiveness. Each material class offers different advantages depending on the therapy being delivered and the destination within the body. Size determines fate. Particles smaller than 10 nanometers are typically cleared quickly by the kidneys, swept out in urine before they can reach their target. Particles larger than 200 nanometers are often captured by immune cells in the liver and spleen, removed as foreign debris. The useful range for most therapies sits between 10 and 100 nanometers, a window where particles can circulate long enough to reach diseased tissue but remain small enough to penetrate barriers that would block larger objects. When a nanoparticle encounters a cell, several things can happen. It might adhere to the cell surface, interacting with receptors or simply sticking through electrostatic forces. It might be engulfed, drawn into the cell through a process called endocytosis, where the cell membrane wraps around the particle and pulls it inward. Once inside, the particle may release its cargo, a drug or genetic material, into the cell's interior. The timing and location of this release can be controlled through careful engineering, a fact that has transformed the concept of targeted delivery. Targeted delivery is the central promise of nanoparticle therapy. Traditional drugs disperse throughout the body, affecting healthy and diseased tissue alike. Chemotherapy damages rapidly dividing cancer cells but also harms bone marrow, digestive lining, and hair follicles. Nanoparticles offer a different approach. By directing treatment specifically to tumors, inflamed tissues, or infected cells, they concentrate therapeutic effects where needed and spare the rest. This is not a perfect process, but even partial targeting can reduce side effects and improve outcomes. The idea is not entirely new. Scientists observed in the 1960s that certain viruses and particles accumulated preferentially in tumors, a clue that the diseased tissue itself might draw in foreign material. Decades of refinement followed, as researchers learned to control particle size, surface chemistry, and payload release. The field moved from observation to engineering, from curiosity to clinical trials. Along the way, one surprising fact emerged. The protein corona, a layer of proteins that coats nanoparticles within seconds of entering the bloodstream, was once seen as a nuisance. It changes the particle's identity, masking design features and often speeding removal by immune cells. Yet researchers discovered that the corona itself could be engineered, seeded with specific proteins to enhance targeting or extend circulation time. What seemed like a flaw became a tool. Nanoparticle therapy matters because it addresses a fundamental challenge in medicine. Many promising drugs fail not because they lack potency, but because they cannot reach their target in sufficient concentration without causing harm elsewhere. Nanoparticles decouple toxicity from efficacy, at least partially, by changing where and when a drug acts. This opens possibilities for treatments that would otherwise be too dangerous or ineffective to use. Let's move to the mechanisms that make this targeting possible. Part 2. Mechanisms of delivery. Nanoparticles reach diseased tissue through several pathways, some passive and some requiring deliberate design. The most studied passive mechanism is the enhanced permeability and retention effect, often abbreviated as EPR. Tumors grow quickly, and their blood vessels form in haste. Unlike healthy capillaries, which have tight junctions between endothelial cells, tumor vessels are leaky. Gaps form between cells, sometimes hundreds of nanometers wide. Nanoparticles slip through these gaps and accumulate in the tumor's interstitial space. Additionally, tumors often lack functional lymphatic drainage, so particles that enter tend to stay. The EPR effect allows nanoparticles to concentrate in tumors without any targeting ligand attached, simply by exploiting the architecture of diseased tissue. The EPR effect works best in animal models, where tumors are uniform and highly vascularized. In human patients, results have been more variable. Tumor heterogeneity, differences in vessel leakiness, and the presence of dense stromal tissue can all limit passive accumulation. This has led researchers to develop active targeting strategies, where nanoparticles carry molecules on their surface that bind to specific receptors overexpressed on cancer cells or tumor blood vessels. Common targeting ligands include antibodies, peptides, and small molecules like folic acid, which binds to folate receptors abundant on many cancer types. Active targeting adds specificity, but it also adds complexity. The ligand must remain functional despite the protein corona. It must bind strongly enough to capture the particle, but not so strongly that it prevents the particle from being internalized. The density of ligands on the particle surface matters, as does the orientation. Too few ligands and binding is weak. Too many and the particles clump together or are recognized as foreign. Optimization requires careful experimentation and is often specific to the cancer type and even the individual patient's tumor biology. Beyond passive and active targeting, a third class of nanoparticles responds to stimuli. These systems remain inert during circulation but release their cargo when triggered by conditions unique to diseased tissue. pH-sensitive particles exploit the acidic environment inside tumors or the even lower pH within cellular compartments called endosomes. As the particle encounters acid, chemical bonds break and the drug is released. Enzyme-responsive systems use proteases, enzymes that tumors secrete to remodel surrounding tissue, as triggers to cleave linkers and free the payload. Heat-activated particles release cargo when external energy, often from focused ultrasound or near-infrared light, raises the local temperature. Light-activated systems use photosensitive molecules that change shape or generate reactive oxygen species when exposed to specific wavelengths, a strategy under exploration for accessible tumors near the skin surface. The cargo itself varies widely. Small molecule drugs, the traditional domain of chemotherapy, are the most common. Encapsulating them in nanoparticles can improve solubility, protect them from degradation, and control release kinetics. RNA-based therapies, including messenger RNA and small interfering RNA, are fragile and require protection from enzymes in the bloodstream. Lipid nanoparticles, now famous for their role in COVID-19 vaccines, shield RNA and facilitate its delivery into cells. CRISPR gene editing systems, which include both guide RNA and Cas enzymes, are large and complex, but nanoparticles have successfully delivered them to specific tissues in preclinical studies. Proteins and peptides, once limited by their instability and poor membrane permeability, are now being packaged into nanoparticles for targeted delivery in cancer immunotherapy and other applications. Barriers remain. The immune system clears foreign particles with remarkable efficiency. Macrophages in the liver and spleen recognize and engulf nanoparticles, especially those coated with certain proteins. The protein corona itself can act as an opsonin, marking particles for destruction. To evade clearance, researchers coat nanoparticles with polyethylene glycol, a polymer that forms a hydrophilic brush layer, reducing protein binding and extending circulation time. This strategy, called pegylation, has been used in approved drugs for decades, though recent studies suggest that repeated dosing can trigger anti-PEG antibodies, accelerating clearance of subsequent doses. Tissue penetration poses another challenge. Even when nanoparticles accumulate in a tumor, they often remain trapped in the perivascular space, unable to diffuse through dense extracellular matrix or reach distant cells. Strategies to improve penetration include using smaller particles, adding enzymes to degrade matrix components, or applying external forces like ultrasound to push particles deeper into tissue. Each approach has trade-offs, and no single solution works universally. We'll continue by exploring the history of how these ideas moved from theory to clinical reality. Part 3. From Laboratory to Clinic. The path from concept to patient is long, and nanoparticle therapy traveled it in stages across several decades. The earliest clinical success came not from cancer treatment but from a more modest application. In the 1990s, a liposomal formulation of the antifungal drug amphotericin B received approval for treating severe fungal infections. Amphotericin B is highly effective but also highly toxic to kidneys when administered in its free form. Encapsulating it in lipid nanoparticles reduced toxicity substantially while preserving antifungal activity. The drug could be given at higher doses and for longer durations, improving outcomes in patients with compromised immune systems. This was proof that nanoparticle delivery could solve real clinical problems. Cancer therapy followed shortly after. Doxil, a liposomal formulation of the chemotherapy drug doxorubicin, gained approval in the mid-90s for treating ovarian cancer and later for Kaposi's sarcoma and other malignancies. Doxorubicin is a potent agent but notorious for causing heart damage when used at cumulative high doses. The liposomal version reduced cardiac toxicity significantly, allowing patients to receive more treatment cycles. It also demonstrated passive tumor targeting through the EPR effect, with imaging studies showing preferential accumulation in tumors compared to free doxorubicin. Doxil remains in use, a testament to the durability of well-designed nanoparticle systems. These early successes shared common features. Both used lipid-based nanoparticles, a relatively simple and biocompatible platform. Both addressed clear clinical needs where toxicity limited the use of effective drugs. Neither required complex targeting ligands or stimuli-responsive release mechanisms. They worked through straightforward encapsulation and modest improvements in biodistribution. This conservative approach made regulatory approval more feasible, as the particles themselves were composed of materials already deemed safe for human use. The next wave of approvals brought more sophisticated designs. Abraxane, approved in the mid-2000s for breast cancer and later for lung and pancreatic cancers, uses a different strategy. Instead of liposomes, it employs albumin nanoparticles to carry paclitaxel, a chemotherapy drug with very poor water solubility. Paclitaxel is normally dissolved in a toxic solvent that causes severe allergic reactions in many patients. Abraxane eliminates the solvent by binding paclitaxel to albumin, a naturally occurring blood protein. The resulting nanoparticles dissolve easily in saline and avoid the hypersensitivity reactions associated with the conventional formulation. Additionally, albumin may enhance tumor uptake through interactions with receptors on cancer cells and endothelial cells, though this mechanism remains debated. Iron oxide nanoparticles took a different route. Initially developed as contrast agents for magnetic resonance imaging, they also showed potential for therapy. When exposed to an alternating magnetic field, iron oxide particles generate heat, a property that can be used to kill cancer cells through hyperthermia. In Europe, a system called Nanotherm received approval in the 2000s for treating glioblastoma, an aggressive brain cancer. The particles are injected directly into the tumor, and then an external magnetic field heats them to temperatures lethal to cells. The treatment is highly localized, sparing surrounding brain tissue. While not widely adopted, it demonstrated that nanoparticles could serve as both delivery vehicles and therapeutic agents in their own right. Regulatory pathways for nanoparticle drugs have evolved as the field matured. Early formulations were often evaluated as modified versions of existing drugs, a classification that simplified approval but limited how novel the designs could be. As more complex systems emerged, regulators developed specific guidance for characterizing nanoparticles. Size distribution, surface charge, stability, and release kinetics must all be measured and controlled within tight specifications. Batch-to-batch consistency is critical, as small changes in manufacturing can alter how particles behave in the body. These requirements add cost and complexity to development, but they ensure that approved products are reproducible and safe. The history also includes setbacks. Several promising nanoparticle formulations failed in late-stage trials despite strong preclinical data. Reasons varied but often involved unexpected immune responses, insufficient tumor accumulation, or marginal improvements over existing therapies that did not justify higher costs. One lesson learned is that animal models, particularly mice with transplanted tumors, do not always predict human outcomes. Mouse tumors tend to be more uniform, more vascularized, and more permeable to nanoparticles than human cancers, which are often heterogeneous and less accessible. This has prompted efforts to develop better preclinical models, including patient-derived xenografts and engineered tissues that more closely mimic human disease. Another lesson concerns the protein corona. Early studies often ignored it, assuming that designed surface features would dominate particle behavior. In reality, the corona forms almost instantly and can completely obscure those features. Researchers now study the corona systematically, identifying which proteins bind under different conditions and how those proteins affect targeting and clearance. Some groups even pre-coat particles with specific corona compositions to control their fate, turning an obstacle into a design element. The clinical history of nanoparticle therapy is one of gradual refinement. Each generation of products built on the successes and failures of the previous one, incorporating new materials, new targeting strategies, and deeper understanding of biological barriers. The field has moved from simple encapsulation to engineered systems that respond to their environment and interact with specific cellular pathways. This progression continues as we look at recent breakthroughs in cancer treatment. Part 4: Recent Advances in Cancer Therapy. The past decade has brought nanoparticle therapy closer to the ideal of truly personalized medicine, with several developments standing out for their clinical impact and mechanistic innovation. Among the most significant is the application of lipid nanoparticles to deliver RNA-based therapies, a technology that gained widespread attention during the COVID-19 pandemic but has equally important implications for cancer treatment. Messenger RNA, or mRNA, can instruct cells to produce therapeutic proteins, including tumor antigens that train the immune system to recognize cancer. The challenge has always been delivery. Naked RNA degrades rapidly in the bloodstream and cannot cross cell membranes efficiently. Lipid nanoparticles solve both problems by encapsulating the RNA in a protective shell and fusing with cell membranes to release the cargo inside. Cancer vaccines using this approach are now in clinical trials for melanoma, pancreatic cancer, and other malignancies. Early results suggest that the vaccines can generate robust immune responses against tumor-specific antigens, and when combined with checkpoint inhibitors, they may improve outcomes in patients who would otherwise have limited options. Prostate cancer has been a particular focus for nanoparticle innovation, driven by the disease's prevalence and the limitations of current treatments. Advanced prostate cancer often becomes resistant to hormonal therapies, and chemotherapy has modest efficacy with significant side effects. Researchers have developed nanoparticles that target prostate-specific membrane antigen, or PSMA, a protein highly expressed on prostate cancer cells. These particles carry chemotherapy drugs, radioactive isotopes, or immune-modulating agents directly to tumors, increasing local drug concentration while reducing systemic exposure. One notable system uses gold nanoparticles conjugated to RNA molecules that silence genes essential for cancer cell survival. The particles are coated with ligands that bind PSMA, ensuring selective uptake by prostate cancer cells. Once inside, the RNA is released and interferes with protein production, triggering cell death. Preclinical studies showed dramatic tumor shrinkage in mouse models, and early-phase human trials have begun. The treatment is given intravenously, and imaging studies confirm that the particles accumulate preferentially in tumors. Side effects have been mild so far, though longer follow-up is needed to assess durability of response. Another approach combines nanoparticles with focused ultrasound to overcome one of the most difficult barriers in treating prostate cancer and brain metastases. The blood-brain barrier and the blood-prostate barrier both limit drug penetration, protecting these tissues from toxins but also from therapies. Focused ultrasound can temporarily disrupt these barriers by generating microbubbles that push apart endothelial cells. When timed with nanoparticle administration, this allows much higher drug concentrations to reach otherwise inaccessible tumors. Clinical trials are underway for both prostate cancer and glioblastoma, with preliminary results showing that the combination is safe and increases drug delivery several-fold compared to nanoparticles alone. Immunotherapy has also benefited from nanoparticle delivery. Checkpoint inhibitors, drugs that release brakes on the immune system, have revolutionized cancer treatment but work in only a subset of patients and can cause severe autoimmune side effects. Nanoparticles loaded with checkpoint inhibitors and delivered directly to tumors may enhance efficacy while reducing systemic toxicity. Some systems go further, carrying not just checkpoint inhibitors but also molecules that stimulate immune cells or reprogram the tumor microenvironment to make it more hospitable to immune attack. These combination particles are being tested in melanoma, lung cancer, and other immunotherapy-responsive malignancies. CRISPR gene editing, delivered by nanoparticles, represents a frontier in cancer treatment. The technology allows precise modification of DNA, potentially correcting mutations that drive cancer or enhancing immune cells to better attack tumors. Lipid nanoparticles have successfully delivered CRISPR components to liver tumors in early human trials, demonstrating that gene editing can occur in vivo with acceptable safety profiles. Extending this to other cancers requires overcoming targeting challenges, as most tissues are less accessible than the liver, but progress is steady. A quieter but equally important advance involves combination therapies. Cancer cells develop resistance to single agents through multiple pathways, so using several drugs simultaneously often works better than sequential treatment. Nanoparticles can carry multiple drugs in precise ratios, ensuring that cells are exposed to the full combination rather than just whichever drug penetrates best. Some systems layer drugs within the particle so they release sequentially, first sensitizing cells and then delivering a lethal dose. Clinical trials of dual-drug nanoparticles have shown improved response rates in breast cancer and ovarian cancer compared to giving the same drugs separately, though the benefit varies depending on the cancer subtype and prior treatments. Imaging and therapy have begun to merge through theranostic nanoparticles, which both diagnose and treat disease. These particles carry imaging agents like fluorescent dyes or radioactive tracers alongside therapeutic cargo, allowing real-time monitoring of where the treatment goes and how tumors respond. Doctors can adjust dosing or switch strategies based on what the imaging reveals, moving toward genuinely adaptive treatment plans. The approach is still largely experimental but has shown promise in trials for liver cancer and certain neuroendocrine tumors. Despite these advances, translation from preclinical success to clinical benefit remains slow and uncertain. We turn next to the challenges that continue to limit the field. Part 5: Persistent Challenges. Even as nanoparticle therapy advances, fundamental obstacles remain. The first and perhaps most humbling is efficiency. In most cases, only a small fraction of administered nanoparticles actually reach the tumor. Studies using radio-labeled particles to track biodistribution consistently show that the majority end up in the liver and spleen, where resident macrophages clear them from circulation. Estimates vary, but even well-designed systems often deliver less than 1% of the injected dose to the target tissue. This means that to achieve therapeutic concentrations in a tumor, very large total doses must be given, raising costs and increasing the risk of off-target effects. The protein corona contributes to this inefficiency. Within seconds of entering the bloodstream, nanoparticles adsorb a layer of plasma proteins that can be several nanometers thick. The composition of this corona depends on particle surface chemistry, size, charge, and the individual patient's protein profile. Once formed, the corona determines how the immune system and other cells perceive the particle. If the corona contains opsonins, proteins that mark foreign material for destruction, macrophages will quickly remove the particle. If it contains disopsonins, proteins that inhibit recognition, the particle may circulate longer. Researchers can engineer particles to attract favorable corona compositions, but this requires detailed knowledge of protein interactions and often fails to translate across species. A particle that evades clearance in mice may be rapidly removed in humans due to differences in plasma protein concentrations and immune activity. Tumor heterogeneity poses another layer of difficulty. Even within a single patient, cancer cells vary genetically and phenotypically. Some cells may overexpress the receptor being targeted, while others express it weakly or not at all. Blood vessel density and permeability differ between the tumor core and its periphery. Stromal cells, extracellular matrix, and immune infiltrates create physical barriers that vary from region to region. A nanoparticle system optimized for one part of the tumor may perform poorly in another. This heterogeneity helps explain why treatments that shrink tumors initially often fail to eradicate them completely. Resistant subpopulations survive, repopulate the tumor, and sometimes develop new resistance mechanisms. Manufacturing scalability remains a practical barrier. Many nanoparticle formulations are synthesized in small batches using methods that work well in the laboratory but are difficult to scale to commercial production. Achieving consistent size distributions, stable encapsulation of drugs, and reproducible surface properties across thousands of liters requires sophisticated equipment and tight process control. Small deviations can change how particles behave in the body, so regulatory agencies demand rigorous quality assurance. These requirements increase development costs and timelines, and some promising formulations have been abandoned simply because they could not be manufactured reliably at scale. Immunogenicity, the tendency to provoke immune responses, is a double-edged issue. Some degree of immune recognition is inevitable with foreign particles, and in certain cases it is desirable, as with cancer vaccines designed to stimulate immunity. In other cases, immune activation is harmful. Complement proteins can bind to nanoparticles and trigger inflammation, sometimes leading to infusion reactions or even anaphylaxis. Repeated dosing can induce antibodies against the particles themselves, particularly if they are coated with polyethylene glycol. These anti-IgG antibodies accelerate clearance of subsequent doses and reduce efficacy. Researchers are exploring alternative coatings, such as spheroionic polymers or self-peptides that mimic the body's own molecules, but each brings its own set of challenges and unknowns. Penetration into solid tumors is limited not just by physical barriers but by abnormal physiology. Tumors often have high interstitial fluid pressure due to leaky vessels and poor lymphatic drainage. This pressure opposes the movement of particles from blood vessels into tissue. Additionally, the chaotic vasculature creates regions of hypoxia, low oxygen concentration, which makes cells less sensitive to many therapies and alters metabolism in ways that can protect cancer cells. Nanoparticles accumulate preferentially in well-perfused regions near vessels but struggle to reach hypoxic zones deeper in the tumor. Strategies to normalize tumor vasculature, such as low-dose antiangiogenic drugs, can improve nanoparticle delivery by reducing vessel leakiness and lowering interstitial pressure, but the optimal timing and combination remain subjects of active investigation. Patient variability complicates efforts to predict who will benefit from nanoparticle therapy. Differences in body composition, metabolism, immune status, and prior treatments all influence how particles distribute and how tumors respond. Some patients clear nanoparticles rapidly, others slowly. Some tumors have highly permeable vessels, others do not. Biomarkers that could guide patient selection are largely lacking. Without reliable predictors, treatments are often given empirically, and response rates in trials reflect this uncertainty. Personalized approaches, where particle formulations are tailored to individual tumor characteristics, are conceptually appealing but currently impractical due to cost and complexity. Regulatory frameworks, while necessary, add layers of difficulty. Because nanoparticles are more complex than small molecule drugs, their characterization requires additional assays and expertise. Agencies require detailed information on particle size distribution, surface chemistry, stability under various conditions, drug loading and release kinetics, and interactions with biological components. Any change in manufacturing process, even minor ones, may necessitate new studies to demonstrate equivalence. These requirements protect patients but also lengthen development timelines and increase costs, which can deter investment in all but the most commercially promising applications. The challenges are not insurmountable, but they demand patience and incremental progress. As researchers address these obstacles, attention must also turn to questions of safety and long-term effects. Part 6: Safety Considerations and Long-Term Effects. Safety is paramount in any medical intervention, and nanoparticle therapies introduce considerations distinct from traditional drugs. The small size and high surface reactivity of nanoparticles mean they interact with biological systems in ways that bulk materials do not. Understanding these interactions requires careful study, both in preclinical models and through vigilant monitoring of patients in trials and post-approval use. Acute toxicity is the most immediate concern. When nanoparticles are administered intravenously, they encounter blood components first. Interactions with platelets, clotting factors, or complement proteins can trigger adverse events ranging from mild infusion reactions to severe thrombosis or complement activation-related pseudoallergy. Symptoms may include flushing, back pain, shortness of breath, and in rare cases, cardiovascular collapse. Premedication with antihistamines and corticosteroids can mitigate some reactions, and slow infusion rates reduce the likelihood of overwhelming the system, but not all reactions are predictable. Careful dose escalation in early phase trials helps identify safe administration protocols before larger studies begin. Organ-specific toxicity depends on where nanoparticles accumulate. The liver and spleen, as primary sites of clearance, are exposed to the highest concentrations. Most lipid and polymer-based nanoparticles are well tolerated by these organs, as they are eventually broken down into components that the body can metabolize or excrete. Metallic nanoparticles, particularly those containing heavy metals, raise more concern. Gold is generally considered inert and non-toxic, but long-term accumulation has not been studied extensively in humans. Iron oxide particles are metabolized through normal iron pathways, but excessive iron can promote oxidative stress and damage cells. Doses are kept well below thresholds expected to cause harm, and imaging studies confirm that most metallic particles are eventually cleared, though the timelines can extend to months or even years. The kidneys filter small particles and molecules, and nanoparticles below a certain size threshold are excreted in urine. This is generally safe, but very high doses or particles that aggregate in the kidneys could potentially cause damage. Renal function is monitored closely in trials involving nanoparticles, and formulations are designed to avoid sizes and surface properties that favor renal accumulation. Similarly, the lungs can be affected if nanoparticles are inhaled or if intravenous particles embolize in pulmonary capillaries. Aggregation is a key risk factor, as clumps of particles can physically block small vessels. Formulations are tested for stability to ensure they remain dispersed under physiological conditions. Long-term effects are harder to assess because they require years of follow-up. Nanoparticles that persist in tissues could theoretically cause chronic inflammation, fibrosis, or other delayed toxicities. Most biodegradable formulations are designed to break down within days to weeks, minimizing this risk. Non-degradable particles, such as certain gold or silica nanoparticles, may remain in the body indefinitely. Animal studies have not shown significant long-term harm from these materials at therapeutic doses, but extrapolating to humans across decades requires caution. Post-market surveillance and long-term registries for patients treated with nanoparticle therapies are essential to detect rare or delayed adverse effects that would not appear in trials lasting months to a few years. Genotoxicity and carcinogenicity are evaluated in standard preclinical assays. Nanoparticles could potentially damage DNA through oxidative stress, direct interaction with genetic material, or interference with repair mechanisms. Most clinically used formulations show no genotoxic effects in these assays, but the testing is not exhaustive and may not capture all mechanisms. The irony of a cancer therapy causing cancer is not lost on regulators, who require extensive data before approving any new formulation. To date, no approved nanoparticle cancer therapy has been convincingly linked to secondary malignancies, though the possibility is monitored. Reproductive and developmental toxicity is another area of concern, particularly for younger patients or those of childbearing potential. Nanoparticles could theoretically cross the placenta or affect germ cells, though most do not. Preclinical studies in pregnant animals are required before trials include such populations, and contraception is typically mandated during treatment and for a period afterward. Data on outcomes in patients who became pregnant after nanoparticle therapy are limited, so recommendations err on the side of caution. Immune-mediated toxicities can be subtle. Beyond acute hypersensitivity reactions, nanoparticles may modulate immune function in ways that become apparent only after prolonged use. Immunosuppression could increase susceptibility to infections or reduce vaccine efficacy. Conversely, immune activation could exacerbate autoimmune conditions or trigger new ones. Patients with pre-existing immune disorders are often excluded from early trials, and immune function is monitored through blood tests for markers of inflammation and immune cell populations. Real-world use in more diverse populations will reveal whether these concerns are significant in practice. Environmental and occupational safety also merit attention. During manufacturing, workers may be exposed to nanoparticle aerosols or skin contact. Inhalation of certain nanoparticles, particularly those that do not dissolve readily, could pose respiratory risks similar to those seen with asbestos or silica dust. Protective equipment and engineering controls minimize exposure, and regulatory guidelines address workplace safety for nanomaterial handling. Disposal of unused or expired nanoparticle formulations must be managed carefully to prevent environmental release, as the ecological effects of many nanomaterials are not fully understood. Patient education plays a role in safety. Individuals receiving nanoparticle therapies need to understand potential side effects, what symptoms warrant immediate medical attention, and any lifestyle modifications that might reduce risk. Clear communication from healthcare providers, supported by written materials and decision aids, helps patients make informed choices and recognize problems early. Safety monitoring does not end with approval. Pharmacovigilance systems track adverse events in real-world use, and regulatory agencies can mandate label changes, restrict use, or withdraw products if new safety signals emerge. The relatively small number of patients treated with any given nanoparticle therapy so far means that rare events may not yet have been observed. As use expands, the safety profile will continue to evolve, and ongoing vigilance remains essential. With safety frameworks in place, the question becomes how nanoparticle therapies are regulated and brought to patients. Part 7. Regulatory Pathways and Approval. Bringing a nanoparticle therapy from laboratory to clinic requires navigating regulatory processes designed to ensure that products are safe, effective, and manufactured consistently. These processes are rigorous and often lengthy, reflecting the complexity of nanoparticle systems and the caution appropriate when introducing new medical technologies. In the United States, the Food and Drug Administration oversees approval through its Center for Drug Evaluation and Research, or, for certain applications, the Center for Biologics Evaluation and Research. The pathway a nanoparticle therapy follows depends on its classification. If it is a reformulation of an existing drug, it may be evaluated as a new drug application with specific guidance for nanoparticle characterization. If it involves novel materials or mechanisms, it may require more extensive preclinical and clinical data. Biologics, such as nanoparticle-delivered gene therapies, follow the Biologics License Application pathway, which has distinct requirements for manufacturing and quality control. Early in development, sponsors typically meet with the agency to discuss the development plan. These pre-investigational new drug meetings help clarify what studies will be needed and what endpoints will be considered meaningful. For nanoparticle therapies, key topics include characterization methods, choice of animal models, dosing strategies, and how to assess biodistribution and pharmacokinetics. The agency may request additional studies if it has concerns about a particular formulation or if similar products have shown unexpected toxicities. Preclinical studies form the foundation of the regulatory package. In vitro assays assess how nanoparticles interact with cells, whether they are toxic at various concentrations, and whether they release their cargo as intended. Animal studies evaluate pharmacokinetics, biodistribution, efficacy in disease models, and safety across multiple organs and dose levels. Good laboratory practice standards ensure that studies are conducted and documented in ways that make the data reliable and reproducible. Toxicology studies in 2 species, typically rodents and non-rodents, are standard, with durations ranging from weeks to months depending on the intended clinical use. Manufacturing is scrutinized closely. Nanoparticle formulations must be produced under good manufacturing practice conditions, with validated processes that yield consistent quality. Specifications are established for critical attributes such as particle size, polydispersity, drug loading, surface charge, endotoxin levels, and sterility. Analytical methods must be validated to show they accurately and precisely measure these attributes. Stability studies demonstrate that the product remains within specifications over its shelf life under recommended storage conditions. Any change to the manufacturing process, even seemingly minor adjustments, may require bridging studies to show that the new batches are equivalent to those used in clinical trials. Clinical trials proceed in phases. Phase 1 studies enroll small numbers of patients, often those with advanced disease who have exhausted other options, to assess safety and determine the maximum tolerated dose. These trials are not primarily designed to measure efficacy, though signals of activity are noted. Phase 2 trials test the product in larger groups to gather preliminary efficacy data and further characterize safety. Phase 3 trials are randomized controlled studies comparing the new therapy to standard of care or placebo, generating the definitive evidence needed for approval. Each phase requires detailed protocols, informed consent documents, institutional review board approval, and oversight to protect participants. For nanoparticle therapies, clinical trial design must account for the unique properties of the product. Pharmacokinetic studies measure not just drug levels in blood but also, when feasible, nanoparticle concentrations and biodistribution. Imaging studies using labeled particles can confirm that they reach the intended target. Biomarkers of response, such as tumor shrinkage on scans or changes in circulating tumor markers, are tracked carefully. Adverse events are categorized and graded, with special attention to novel toxicities that might be specific to nanoparticles. Regulatory approval is granted if the data demonstrate a favorable benefit-risk profile. The bar for approval depends on the indication. For a disease with no effective treatment, even modest efficacy with acceptable safety may be sufficient. For a disease with multiple existing therapies, the new product must offer clear advantages, such as better efficacy, fewer side effects, more convenient administration, or benefits in a subpopulation that does not respond to current options. Approval may be conditional or accelerated if the product addresses an unmet need and shows promise in early trials, with the expectation that confirmatory studies will follow. Post-approval, companies must continue to monitor the product. Phase 4 studies may be required to answer questions that were not fully resolved during development. Adverse event reporting systems capture problems that occur in real-world use, where patient populations are more diverse than in trials. Manufacturing continues under regulatory oversight, with periodic inspections to ensure compliance with good manufacturing practices. If safety concerns arise, the agency can require label changes, restrict use to certain patient populations, or withdraw approval. International regulatory pathways are similar in structure but differ in details. The European Medicines Agency evaluates products for use in the European Union, with a centralized procedure for most new drugs. Individual countries may have additional requirements. Regulatory agencies in other regions, such as Japan, Canada, and Australia, have their own processes, and companies seeking global approval must navigate each. Harmonization efforts, such as those led by the International Council for Harmonization, aim to align requirements and reduce duplication, but significant differences remain. Orphan drug designations and breakthrough therapy designations can expedite development for products targeting rare diseases or offering substantial improvements over existing therapies. These designations provide benefits such as extended market exclusivity, reduced application fees, and more frequent interactions with regulators. Several nanoparticle therapies have received such designations, reflecting the potential they hold for difficult-to-treat conditions. The regulatory landscape continues to evolve as experience with nanoparticle products grows. Agencies refine their guidance based on lessons learned, and new frameworks are developed for emerging areas such as personalized nanoparticle therapies or combination products that integrate diagnostics and treatment. Navigating this landscape requires expertise, resources, and perseverance, but it is through this process that innovations reach the patients who need them. Looking beyond current regulations, the future of nanoparticle therapy depends on scientific advances and their translation into practical treatments. Part 8: Future Directions in Design and Engineering. The next generation of nanoparticle therapies will likely build on lessons learned from current systems while incorporating advances in materials science, biological understanding, and engineering precision. Several directions appear particularly promising, though all remain works in progress with uncertain timelines for clinical translation. One emerging area involves stimuli-responsive systems that release their cargo only when specific biological conditions are met. Current pH-sensitive and enzyme-responsive particles represent early steps in this direction, but future designs may respond to combinations of stimuli, requiring multiple signals before activation. For instance, a particle might remain inert until it encounters both low pH and a specific protease, conditions that occur together in tumors but rarely in healthy tissue. This multigating approach could reduce off-target release and improve the therapeutic window. Researchers are also exploring particles that respond to disease-specific metabolites or changes in redox environment, creating formulations tailored to the biochemical signatures of particular cancers or inflammatory conditions. Another frontier is the integration of artificial intelligence into nanoparticle design. Machine learning algorithms can analyze vast data sets from previous experiments to predict which combinations of size, shape, surface chemistry, and cargo will perform best for a given application. Early studies have used these approaches to optimize formulations more quickly than traditional trial and error methods would allow. As databases grow and models improve, it may become possible to design personalized nanoparticles for individual patients based on their tumor characteristics, immune profile, and genetic makeup. This vision remains distant, but incremental progress is being made as computational tools become more sophisticated and accessible. Self-assembling nanoparticles represent a shift from preformed particles to systems that assemble in situ after administration. These formulations are injected as small molecules or peptides that spontaneously organize into nanoparticles when they encounter physiological conditions. Self-assembly can occur in response to pH changes, temperature shifts, or interactions with specific biomolecules. The advantage is that small precursors may penetrate tissues more easily than preformed particles, assembling only after reaching the target site. This approach is still largely experimental, with challenges in controlling assembly kinetics and ensuring that particles form with the intended properties, but proof-of-concept studies have shown promise in several preclinical models. Biodegradable materials continue to evolve. Current polymers like polylactic-glycolic acid degrade predictably but leave behind acidic byproducts that can cause local inflammation at high concentrations. Newer polymers are being developed with degradation products that are neutral or even beneficial, such as amino acids or natural metabolites. Some researchers are exploring materials derived from biological sources, such as silk proteins or bacterial polyesters, which may offer better biocompatibility and less immune recognition. The challenge is achieving the right balance of stability during storage and circulation with timely degradation once the particle has delivered its cargo. Multifunctional particles that combine diagnosis and therapy, often called theranostics, are becoming more sophisticated. Beyond simply carrying an imaging agent alongside a drug, next-generation systems may monitor treatment response in real time and adjust drug release accordingly. For example, a particle could sense markers of cell death and increase release rates if the tumor is responding or decrease release if resistance develops. Implementing such feedback loops within a nanoparticle requires miniaturized sensors and responsive mechanisms that are only beginning to be realized, but the concept illustrates how far the field might eventually progress. Cell-derived nanoparticles offer a biological alternative to synthetic systems. Exosomes, small vesicles naturally secreted by cells, can be engineered to carry drugs or genetic material. Because they originate from cells, they may evade immune recognition more effectively than synthetic particles. Red blood cell membranes and cancer cell membranes have been used to coat nanoparticles, providing a biological disguise that prolongs circulation and enhances targeting. These biomimetic approaches are appealing but face manufacturing challenges, as isolating and modifying biological materials at scale is complex and expensive. Shape is another parameter under exploration. Most nanoparticles are spherical because spheres are easiest to manufacture and characterize, but non-spherical particles behave differently in the body. Rod-shaped particles may circulate longer and penetrate tumors more effectively than spheres of the same volume. Disc-shaped particles can adhere to vessel walls in ways that spheres cannot, potentially useful for targeting inflamed endothelium. The effects of shape are not fully understood and likely depend on the specific application, but the ability to control particle geometry is improving, opening new design possibilities. Combination therapies delivered by nanoparticles will likely become more common. Instead of single drugs, particles may carry two or more agents that work synergistically, along with molecules that modulate the tumor microenvironment or enhance immune infiltration. Coordinating the release of multiple payloads is complex, but doing so could address resistance mechanisms and improve outcomes. Some systems use layered structures where outer shells release first to prime the tumor, followed by inner cores that deliver the main therapeutic punch. Despite these advances, translation to clinical use remains slow. Many promising technologies fail not because the science is flawed but because they cannot be manufactured reliably, scaled economically, or navigated through regulatory pathways within reasonable timelines. The gap between what is possible in principle and what is practical in reality is wide, and bridging it requires collaboration among scientists, engineers, clinicians, regulators, and industry partners. The future of nanoparticle therapy is not a single breakthrough but a gradual accumulation of improvements. Each small advance in targeting, each incremental reduction in toxicity, and each step toward personalization brings the field closer to its potential. Patience and persistence are essential, as is realistic assessment of what can be achieved in the near term versus what remains speculative. We turn now to how these therapies are actually used in clinical practice and what patients experience. Part 9. Clinical application and patient experience. When a patient receives nanoparticle therapy, the process differs in some respects from conventional treatments, though many elements remain familiar. The decision to use a nanoparticle formulation typically arises when standard therapies have failed, when side effects of conventional drugs are intolerable, or when clinical trial participation offers access to experimental options. Oncologists discuss the rationale, potential benefits, known risks, and uncertainties with patients, ensuring informed consent before proceeding. Preparation for treatment involves baseline assessments. Blood tests measure organ function, immune cell counts, and markers of disease burden. Imaging studies establish the extent of cancer and provide a reference for measuring response. Patients may undergo allergy testing or receive pre-medication with antihistamines and corticosteroids to reduce the risk of infusion reactions. These steps are similar to preparation for many intravenous therapies, but the emphasis on monitoring for novel toxicities specific to nanoparticles adds layers of caution. Administration is usually intravenous, though intratumoral injection is used for some formulations. Infusions are given slowly, often over 1 to several hours, with nurses monitoring vital signs and watching for signs of reactions. The first dose is approached with particular care, as it is most likely to trigger unexpected responses. If no problems arise, subsequent doses may be given more quickly. Some nanoparticle therapies are administered in cycles, with treatment days followed by rest periods to allow recovery from side effects and to prevent cumulative toxicity. Others are given as single doses or short courses, depending on the formulation and indication. Patients generally do not feel the nanoparticles themselves. Unlike some chemotherapies that cause immediate nausea or flushing, many nanoparticle formulations produce little in the way of acute sensations during infusion. Side effects, when they occur, often develop hours to days later. Fatigue is common, as are mild fever and flu-like symptoms, reflecting immune system activation. Nausea, though less severe than with conventional chemotherapy in many cases, can still occur. Injection site reactions, if the formulation is given intratumorally, include pain, swelling, and redness that usually resolve within days. The reduction in certain side effects is one of the main attractions of nanoparticle therapy. Doxil, for example, largely avoids the hair loss and severe nausea associated with free doxorubicin, and the risk of heart damage is lower. Patients appreciate these differences, as quality of life during treatment is a significant concern. However, nanoparticle formulations can introduce their own side effects. Doxil is associated with hand-foot syndrome, a painful skin reaction on palms and soles that occurs more frequently than with conventional doxorubicin. This illustrates that improving one aspect of a therapy may create new challenges elsewhere. Monitoring during and after treatment is more intensive than with some traditional drugs. Because nanoparticles distribute differently and may have delayed or prolonged effects, follow-up visits track not just tumor response but also particle-specific concerns. Blood tests check liver and kidney function, complete blood counts assess for bone marrow suppression, and immune panels may be drawn if immune modulation is expected. Imaging studies, sometimes using the same nanoparticles labeled with tracers, confirm that particles are reaching tumors and document changes in tumor size or metabolic activity. Response to therapy varies widely. Some patients experience dramatic tumor shrinkage and prolonged remission, while others see little benefit. Predicting who will respond remains difficult, as biomarkers for nanoparticle therapy responsiveness are not well established. Tumor characteristics such as vascularity, receptor expression, and immune infiltration likely play roles, but these are not routinely measured in ways that guide treatment decisions. Researchers are working to identify predictive markers, but until they are validated, treatment decisions rely on clinical judgment and often trial and error. Patients enrolled in clinical trials face additional uncertainties. They may be randomized to receive the experimental nanoparticle therapy or a control treatment, and they will not know which until the trial ends. They undergo more frequent monitoring and testing than would occur in standard care, which can be burdensome but also reassuring. The hope of accessing a potentially superior treatment motivates many to participate, as does the desire to contribute to medical progress. Informed consent documents lay out the risks and unknowns, but translating statistical probabilities and theoretical concerns into personal decisions is never straightforward. Cost is a factor, though it is often opaque to patients in systems where insurance or public health care covers treatment. Nanoparticle therapies tend to be expensive due to complex manufacturing and the need to recoup development costs. In some cases, insurers may balk at covering newer formulations if conventional alternatives exist, even if the newer option has advantages. Prior authorization processes and appeals can delay access, adding stress during an already difficult time. For patients without adequate coverage, out-of-pocket costs can be prohibitive, and assistance programs or clinical trial enrollment may be the only routes to treatment. Psychological aspects of receiving an experimental or novel therapy should not be underestimated. Some patients feel hopeful, seeing nanoparticle therapy as cutting-edge and evidence of progress in the fight against their disease. Others feel anxious about unknowns, worrying that not enough is understood about long-term effects or that they are being used as guinea pigs. Open communication with healthcare teams, access to clear information, and support from counselors or patient advocacy groups can help individuals navigate these emotions. The experience of caregivers and family members also matters. They attend appointments, manage medications, watch for side effects, and provide emotional support. When a patient is on a novel therapy, caregivers may feel the weight of uncertainty alongside the patient. Education about what to expect and when to seek help empowers them to contribute effectively to care. Post-treatment life for those who respond well involves ongoing surveillance for recurrence and monitoring for late effects. Patients may undergo periodic scans and blood tests for months to years, gradually transitioning to less frequent follow-up if they remain disease-free. For those whose cancer progresses despite nanoparticle therapy, the focus shifts to subsequent treatment options, symptom management, and palliative care. The decision to stop active treatment is never easy, and the hope that nanoparticle therapy represented can make the transition emotionally difficult. Real-world evidence, collected from patients treated outside of clinical trials, is beginning to supplement trial data. Registries and databases track outcomes, side effects, and patterns of use across diverse populations. This information helps refine understanding of who benefits and under what circumstances, gradually building a more complete picture than trials alone can provide. Nanoparticle therapy is not a panacea, and patient experiences reflect the full spectrum from remarkable success to disappointing failure. Each individual's journey through diagnosis, treatment, and follow-up is unique, shaped by biology, circumstance, and the choices available at each juncture. With clinical realities in mind, we now consider how costs and accessibility shape who can benefit from these advances. Part 10: Economic and Access Considerations. The promise of nanoparticle therapy must be weighed against its cost, a reality that influences which therapies are developed, approved, and ultimately made available to patients. Developing a new nanoparticle formulation from concept to approval can require hundreds of millions of dollars and a decade or more of effort. These costs arise from preclinical research, manufacturing process development, clinical trials, regulatory submissions, and post-approval commitments. Companies invest with the expectation of eventual profit, which necessitates pricing that recovers development costs and generates returns for shareholders. Manufacturing nanoparticles is inherently more complex and expensive than producing traditional small molecule drugs. Precise control over particle size, uniformity, and drug loading requires specialized equipment and expertise. Clean room facilities meeting good manufacturing practice standards are costly to build and operate. Quality control testing for nanoparticle formulations is extensive, involving techniques such as dynamic light scattering, electron microscopy, and stability studies under various conditions. Each batch must meet strict specifications, and any deviation can result in rejection, adding to costs. Unlike small molecules that can often be synthesized in bulk and then portioned out, nanoparticles are typically produced in controlled batches, limiting economies of scale. These manufacturing realities translate into high prices for approved nanoparticle therapies. Doxil, for instance, has historically been priced significantly higher than conventional doxorubicin, despite containing the same active drug. The premium reflects the cost of the liposomal delivery system, its development, and the value of reduced toxicity. Abraxane similarly costs more than solvent-based paclitaxel. Insurers and healthcare systems must decide whether the benefits justify these price differences, a calculus that varies by region and payer. In wealthy countries with robust healthcare systems, most approved nanoparticle therapies are eventually covered by insurance or public health programs, though coverage decisions can be contentious. Health technology assessment agencies evaluate cost-effectiveness, comparing the incremental benefit of the new therapy to its incremental cost relative to existing treatments. If the cost per quality-adjusted life year gained exceeds accepted thresholds, coverage may be denied or restricted to specific patient subgroups. This means that even in resource-rich settings, not all patients who might benefit gain access. The situation is more challenging in low- and middle-income countries, where healthcare budgets are constrained and the infrastructure for administering complex therapies may be limited. Nanoparticle formulations that require refrigeration, specialized infusion equipment, or close monitoring may be impractical in settings where even basic chemotherapy is difficult to deliver reliably. Generic versions of older nanoparticle therapies are beginning to appear, which could lower costs, but quality concerns and regulatory hurdles complicate their adoption. International organizations and partnerships are exploring ways to make advanced cancer treatments more accessible globally, but progress is slow. Intellectual property protections add another layer to the economic landscape. Patents on nanoparticle formulations and manufacturing processes grant companies exclusive rights for years, preventing competitors from entering the market and driving prices down. While patents incentivize innovation by ensuring a return on investment, they also delay the availability of more affordable versions. The balance between rewarding innovation and ensuring access is contentious, with different stakeholders advocating for different priorities. Clinical trials themselves raise access questions. Participation in trials offers patients access to experimental therapies that might otherwise be unavailable, but trial eligibility criteria often exclude individuals with comorbidities, poor performance status, or certain demographics. Geographic location matters as well, with trials concentrated in urban academic medical centers, making participation difficult for those in rural or underserved areas. Efforts to diversify trial populations and expand trial sites are ongoing but face logistical and financial barriers. Value-based pricing models are being explored as alternatives to traditional pricing. Under these models, prices are tied to patient outcomes, with manufacturers receiving higher payments if the therapy proves effective and lower payments if it does not. This aligns incentives and shifts some risk from payers to manufacturers, but implementing such models requires robust outcome tracking and agreement on what constitutes success. Nanoparticle therapies, with their variable efficacy across patients, could be candidates for value-based arrangements, though practical challenges remain. Patient assistance programs offered by manufacturers provide free or reduced-cost drugs to individuals who cannot afford them, but these programs vary in scope and accessibility. Application processes can be cumbersome, and not all who need help qualify. Advocacy groups sometimes assist patients in navigating these programs, but the need for such assistance highlights gaps in the healthcare system. Opportunity costs must also be considered. Money spent on expensive nanoparticle therapies is money not available for other healthcare priorities, such as preventive services, vaccinations, or treatments for more common conditions. Health systems must allocate finite resources across competing needs, and the high cost of cutting-edge therapies can strain budgets in ways that affect other patients. These trade-offs are rarely visible to individuals focused on their own care, but they are real and consequential. Research funding priorities influence which nanoparticle therapies are developed. Diseases affecting large populations in wealthy markets attract more investment than rare diseases or conditions prevalent mainly in poorer regions. This creates disparities in therapeutic innovation, with some patient groups benefiting disproportionately while others are left behind. Public funding and philanthropic support can partially offset market-driven priorities, but they are limited relative to the scale of private investment. The economic and access challenges surrounding nanoparticle therapy are not unique to this field but are shared by many advanced medical technologies. Addressing them requires coordinated efforts among governments, industry, payers, healthcare providers, and patient advocates. Solutions will likely involve a mix of policy interventions, innovative financing mechanisms, technology transfer to lower-cost manufacturing regions, and continued scientific progress that drives down production costs over time. Ensuring that the benefits of nanoparticle therapy reach those who need them, regardless of geography or economic status, is an ongoing struggle. The technical achievements in the laboratory are only part of the story. Translating those achievements into equitable healthcare requires grappling with economic realities and making difficult choices about priorities and resource allocation. As we look at the broader landscape, ethical considerations come into focus. Part 11: Ethical Dimensions. Nanoparticle therapy raises ethical questions that extend beyond the usual concerns of medical research and treatment. The complexity of these systems, their novelty, and their potential for both benefit and harm require careful consideration of how they are developed, tested, and deployed. Informed consent in clinical trials presents particular challenges. Nanoparticle therapies often involve mechanisms that are difficult to explain in lay terms. Patients must understand not just what the treatment aims to do, but also how it differs from conventional approaches and what uncertainties remain. Consent documents can run to many pages, filled with technical details and caveats. The question is whether patients truly grasp what they are agreeing to or whether they sign out of desperation, trust in their physicians, or hope that any experimental option is better than none. Researchers strive for meaningful consent, using visual aids and extended discussions, but the gap between technical reality and patient comprehension is not easily closed. Equipoise, the principle that a trial is ethical only if there is genuine uncertainty about which treatment is better, becomes complicated when experimental nanoparticle therapies are compared to standard care. Researchers and patients may have different views on whether equipoise exists. A researcher might see the evidence as balanced, justifying randomization. A patient might believe the experimental arm offers hope that standard treatment does not, making randomization feel like a barrier to what they perceive as their best chance. This tension is inherent in trial design but is heightened when one arm involves a novel technology that captures public and media attention. Vulnerable populations require special consideration. Cancer patients facing poor prognosis may feel coerced by circumstance into enrolling in trials, even if they have reservations. Ensuring that consent is voluntary and that patients understand they can decline without affecting their standard care is essential. Additionally, disparities in trial access mean that some populations are underrepresented, limiting the generalizability of results and perpetuating inequities. Efforts to include diverse participants must balance the goal of representative data with protections against exploitation of marginalized groups who might be recruited disproportionately due to lack of alternative options. The use of animals in preclinical research is another ethical concern. Developing nanoparticle therapies requires extensive testing in mice, rats, and sometimes larger animals to assess safety and efficacy before human trials begin. These studies involve inducing diseases, administering experimental treatments, and ultimately euthanizing animals for analysis. While regulations mandate humane treatment and the minimization of suffering, the use of animals remains controversial. Alternatives such as organoids and computational models are being developed but cannot yet replace animal studies entirely. Researchers must weigh the potential human benefit against the animal harm involved, a calculation that varies by individual values and societal norms. Dual-use concerns arise when technologies developed for therapy could be repurposed for harm. Nanoparticles designed to deliver drugs could theoretically be adapted to deliver toxins or biological agents. While this risk is not unique to nanomedicine, the precision and targeting capabilities of nanoparticles could make such misuse more effective. Oversight and export controls aim to prevent dual-use technologies from falling into the wrong hands, but the global nature of scientific research makes absolute control difficult. Open publication of methods enables progress but also disseminates knowledge that could be misused. Balancing transparency and security is an ongoing challenge. Environmental ethics also come into play. Nanoparticles released into the environment during manufacturing, disposal, or through patient excretion could accumulate in ecosystems. Their small size allows them to be taken up by organisms and potentially move through food chains. Most biodegradable nanoparticles break down into harmless components, but non-degradable types could persist. The long-term ecological effects are not well studied, and the precautionary principle suggests caution. Regulations governing nanomaterial disposal are evolving, but gaps remain, particularly in regions with less stringent environmental protections. Equity in access, discussed earlier in economic terms, also has ethical dimensions. If nanoparticle therapies offer substantial benefits but are available only to wealthy patients in affluent countries, this deepens global health disparities. The principle of justice in healthcare demands that benefits be distributed fairly, not just according to ability to pay. Pharmaceutical companies operate in competitive markets and must balance profitability with social responsibility. Tiered pricing, voluntary licensing, and partnerships with generic manufacturers are mechanisms to improve access, but they require political will and cooperation across sectors. Paternalism versus patient autonomy is a recurring theme. Physicians may believe they know what is best for a patient based on medical evidence and experience. Patients may prioritize different outcomes, such as quality of life over longevity, or may wish to try experimental therapies despite long odds. Respecting autonomy means honoring patient choices even when they diverge from medical recommendations, provided the patient is informed and competent. Nanoparticle therapies, often positioned as last resort options, test this balance. Physicians must provide honest assessments of likely benefit without crushing hope, while patients must navigate uncertainty and make decisions that align with their values. The concept of benefit itself is complex. Clinical trials measure survival, tumor response, and quality of life, but these do not capture everything that matters to patients. Participating in research may provide a sense of purpose or contribution to future patients, a benefit not reflected in efficacy data. Conversely, the burden of frequent monitoring, travel to trial sites, and side effects may outweigh modest clinical gains. Ethical trial design considers these factors and strives to minimize burden while maximizing information gained. Conflicts of interest in research funding and physician relationships with industry are scrutinized closely. When pharmaceutical companies sponsor trials, there is potential for bias in study design, data interpretation, or publication decisions. Disclosure requirements and independent oversight aim to mitigate these risks, but the influence of funding sources cannot be eliminated entirely. Physicians who receive consulting fees or speaking honoraria from companies may, consciously or unconsciously, favor those companies' products. Transparency and institutional policies help manage conflicts, but they remain a persistent ethical concern. Post-trial access to experimental therapies is another issue. If a nanoparticle therapy proves effective during a trial, participants may wish to continue receiving it after the trial ends. Companies are not always obligated to provide continued access, particularly if the product is not yet approved. Patients who benefited may face the prospect of losing a treatment that was working, raising questions about the ethics of starting a therapy that cannot be sustained. Some trial protocols include provisions for extended access, but this is not universal. The ethical landscape of nanoparticle therapy is not static. As technologies evolve and societal values shift, new questions emerge. Ongoing dialogue among researchers, ethicists, patients, and the public is essential to navigate these complexities and ensure that progress serves the common good. Part 12. Comparisons with other treatment modalities. Nanoparticle therapy does not exist in isolation but is one option among many for treating cancer and other diseases. Understanding its place requires comparing it to established modalities and assessing where it offers advantages and where it falls short. Surgery remains the primary curative treatment for many solid tumors when detected early. Removing a tumor eliminates malignant cells directly and provides tissue for diagnosis and molecular analysis. Nanoparticle therapy cannot replace surgery for localized disease, but it may complement it. Neoadjuvant therapy, given before surgery, can shrink tumors to make resection easier or less extensive. Adjuvant therapy, given after surgery, targets residual disease to reduce recurrence risk. Nanoparticles could improve both approaches by delivering drugs more effectively to residual cancer cells while sparing normal tissue, potentially reducing the need for aggressive surgery or improving outcomes when surgery is not curative. Radiation therapy uses high-energy beams to kill cancer cells by damaging DNA. It is highly effective for localized tumors and can be precisely targeted using modern imaging and delivery techniques. Nanoparticles are being explored as radiosensitizers, substances that make tumors more vulnerable to radiation. Gold nanoparticles, for instance, concentrate in tumors and absorb radiation energy, amplifying the dose delivered to cancer cells while sparing surrounding tissue. This approach is still experimental but could allow lower radiation doses with equivalent or better efficacy, reducing side effects. Nanoparticles carrying radioactive isotopes, a form of targeted radionuclide therapy, deliver radiation directly to cancer cells, combining the precision of targeted delivery with the destructive power of radiation. Conventional chemotherapy, delivered systemically as free drugs, has been the backbone of cancer treatment for decades. Nanoparticle formulations aim to improve chemotherapy by altering biodistribution, reducing toxicity, and sometimes overcoming resistance. In head-to-head trials, nanoparticle versions of drugs like doxorubicin and paclitaxel have shown reduced side effects and, in some cases, improved efficacy. However, the benefits are often incremental rather than transformative. Patients still experience side effects, tumors still develop resistance, and cures remain elusive for most advanced cancers. Nanoparticles enhance chemotherapy but do not fundamentally change its limitations. Immunotherapy has revolutionized treatment for several cancer types by harnessing the patient's immune system to attack tumors. Checkpoint inhibitors, CAR-T cell therapy, and cancer vaccines work through mechanisms entirely different from chemotherapy. Nanoparticles are being integrated into immunotherapy to improve delivery of immune-stimulating molecules, target antigens more effectively, or modulate the tumor microenvironment. Early results suggest that combining nanoparticles with immunotherapy could enhance response rates, particularly in tumors that are otherwise resistant. However, immunotherapy's effectiveness is highly variable, and adding nanoparticles does not guarantee success. The interplay between nanoparticle properties and immune responses is complex and not fully understood. Targeted therapies, small molecule drugs, or antibodies that inhibit specific proteins driving cancer growth offer precision different from nanoparticles. Drugs like imatinib for chronic myeloid leukemia or trastuzumab for HER2-positive breast cancer have changed outcomes dramatically for patients with specific molecular profiles. These therapies work because they block essential pathways in cancer cells. Nanoparticles do not compete with this mechanism but could enhance it by improving drug delivery or combining targeted agents with other therapies. For instance, delivering a kinase inhibitor via nanoparticles might reduce systemic side effects or overcome resistance caused by poor drug penetration into tumors. Hormonal therapies for cancers driven by hormones, such as prostate and breast cancer, are effective for many patients but eventually lead to resistance. Nanoparticles carrying hormonal agents or drugs that block hormone receptors could prolong efficacy or reduce side effects. This application is less explored than chemotherapy delivery but represents a potential niche where nanoparticles add value. Emerging modalities like oncolytic viruses, which infect and kill cancer cells while stimulating immune responses, and photodynamic therapy, which uses light-activated drugs to destroy tumors, intersect with nanoparticle technology. Nanoparticles can deliver oncolytic viruses more effectively or carry photosensitizers that concentrate in tumors, enhancing the precision and efficacy of these approaches. These combinations are largely preclinical but illustrate how nanoparticles can integrate with diverse treatment strategies. In terms of patient experience, nanoparticle therapy often sits between conventional chemotherapy and newer immunotherapies. Side effects are typically milder than traditional chemotherapy but more significant than many targeted therapies. Administration is similar to chemotherapy, requiring intravenous infusion and medical supervision. Response rates vary but are generally comparable to or modestly better than conventional formulations of the same drugs. The promise of dramatically improved outcomes has not yet been realized for most cancers, though specific applications show clearer benefits. Cost comparisons favor conventional chemotherapy in the short term, as generic small-molecule drugs are inexpensive. Nanoparticle formulations are more costly upfront but may reduce overall healthcare costs if they prevent hospitalizations for side effects or allow patients to remain on treatment longer. Cost-effectiveness analyses yield mixed results depending on the specific formulation, cancer type, and healthcare system. In some cases, the added cost is justified by improved outcomes or quality of life. In others, the benefit is insufficient to warrant the expense. Nanoparticle therapy is not a replacement for existing modalities but a complementary tool. Its role will likely expand as formulations improve and as integration with other treatments becomes more sophisticated. The goal is not to choose one modality over another but to use combinations tailored to individual patients and their specific disease characteristics. Part 13: Global Research Efforts and Collaboration. Nanoparticle therapy research is a global endeavor, with contributions from academic institutions, industry, and government agencies across continents. The distribution of effort reflects both scientific capability and economic resources, with most activity concentrated in North America, Europe, and parts of Asia. The United States leads in terms of funding and publication output. The National Institutes of Health, through its National Cancer Institute and other institutes, provides substantial support for nanomedicine research. Academic centers with strong programs in materials science, bioengineering, and oncology drive much of the innovation. Companies ranging from large pharmaceutical firms to small biotech startups translate academic discoveries into clinical candidates. The regulatory pathway through the FDA, while rigorous, is well established, and the large commercial market incentivizes investment. Europe also contributes significantly, with research hubs in the United Kingdom, Germany, France, and the Netherlands. The European Medicines Agency coordinates regulatory approval across member states, and funding comes from national agencies as well as European Union programs that support collaborative research. European researchers have pioneered work on polymeric nanoparticles, stimuli-responsive systems, and theranostic applications. Academic industry partnerships are common, and the proximity of institutions across the continent facilitates collaboration. China has rapidly expanded its nanomedicine research capacity. Government initiatives prioritize nanotechnology as a strategic area, and funding has grown substantially. Chinese institutions publish a large volume of research on nanoparticle synthesis, characterization, and preclinical testing. Clinical translation has been slower, partly due to regulatory frameworks that are still evolving, but several nanoparticle formulations developed in China are in trials. Collaboration between Chinese and Western researchers is increasing, though geopolitical tensions sometimes complicate partnerships. Japan, South Korea, and Singapore are active in nanomedicine, with strengths in materials science and manufacturing. Japanese researchers have contributed to understanding protein corona effects and developing biocompatible materials. South Korea invests heavily in nanotechnology, and its advanced manufacturing capabilities position it well for producing clinical-grade nanoparticles. Singapore serves as a regional hub, with government-supported research institutes attracting international talent. Other regions contribute but face greater challenges. India has growing research activity, particularly in developing cost-effective nanoparticle formulations for diseases prevalent in resource-limited settings. Funding is more constrained, and infrastructure for advanced manufacturing and clinical trials is less developed than in wealthier countries. Latin America, Africa, and parts of the Middle East have pockets of nanomedicine research but are limited by resources and, in some cases, political instability. International collaboration takes many forms. Joint funding programs support multinational research teams. Conferences bring together scientists from around the world to share findings and forge partnerships. Open-access publishing and preprint servers accelerate the dissemination of knowledge, though access to cutting-edge tools and reagents is not uniform. Some institutions in lower-income countries lack resources to replicate experiments or build on published work, creating a divide between those who generate knowledge and those who can apply it. Intellectual property complicates collaboration. Patents filed in one country may not be recognized in another, and disputes over ownership of discoveries can arise when multiple institutions contribute. Licensing agreements and material transfer agreements govern the exchange of nanoparticles, reagents, and technologies, sometimes slowing research if negotiations are protracted. Balancing the need to protect innovations with the desire to share knowledge for the public good remains a persistent tension. Standardization efforts aim to improve reproducibility and comparison across studies. Organizations like the International Organization for Standardization and the National Institute of Standards and Technology develop reference materials and measurement protocols for nanoparticles. Without standardized methods, results from different labs can be difficult to compare, hindering progress. Adoption of standards is gradual, and some researchers resist constraints on their methodologies, but consensus is growing that standardization benefits the field. Part 14. Lessons from Clinical Failures. Not all nanoparticle therapies that enter clinical testing succeed, and examining failures provides insights as valuable as studying successes. Several high-profile candidates have faltered in late-stage trials despite promising preclinical data, revealing gaps in understanding and highlighting challenges that the field continues to address. One instructive case involved a targeted nanoparticle system designed to deliver chemotherapies specifically to tumors overexpressing a particular receptor. Preclinical studies in mice showed impressive tumor shrinkage with minimal toxicity. The particles accumulated in tumors at high concentrations, and the targeting ligand appeared to function as intended. Phase 1 trials in humans confirmed safety and showed some evidence of tumor targeting. However, Phase 3 trials comparing the nanoparticle formulation to standard chemotherapy found no significant improvement in survival or response rates. Post-hoc analysis revealed several problems. Human tumors were more heterogeneous than mouse models, with variable receptor expression even within individual tumors. The protein corona that formed on the particles in human blood differed substantially from that in mice, altering targeting efficiency. Additionally, the chemotherapy drug chosen was already highly effective in its conventional form, leaving little room for improvement through better delivery. Another failure involved a stimuli-responsive nanoparticle designed to release its drug payload only in the acidic environment of tumors. The concept was sound, and laboratory tests showed precise pH-triggered release. Early clinical trials demonstrated that the particles circulated safely and accumulated in tumors. However, efficacy was disappointing. Investigation revealed that while tumors are generally more acidic than normal tissue, the pH difference is modest and variable. Some regions of tumors, particularly necrotic cores, are highly acidic, but viable tumor cells at the periphery exist in near-neutral pH. The particles released drug mainly in areas where cells were already dying, missing the actively proliferating cells that needed to be targeted. This highlighted the importance of understanding the microenvironment in detail and designing systems that respond to conditions where viable cancer cells reside. A metallic nanoparticle platform intended for both imaging and therapy failed due to unexpected immune reactions. Preclinical toxicology studies had not revealed problems, but when scaled to human doses, a subset of patients developed severe complement activation and cytokine release. The reactions occurred despite premedication and were unpredictable, appearing in some patients but not others. The variability suggested individual differences in immune responsiveness that were not captured in animal models or small early-phase trials. The program was halted, and the company never identified the precise mechanism. This case underscored the difficulty of predicting immune responses to novel materials and the need for larger safety databases before advancing to pivotal trials. Manufacturing inconsistencies have derailed other candidates. One polymer-based nanoparticle showed excellent results in Phase 2 trials, but when the company scaled up production for Phase 3, the particles produced in the larger batches had slightly different size distributions and release kinetics. These differences, though within the original specifications, altered biodistribution enough to reduce efficacy. By the time the problem was identified and corrected, significant time and resources had been lost. Regulatory agencies required new studies with the corrected formulation, effectively restarting development. This illustrates how sensitive nanoparticle performance can be to seemingly minor manufacturing variations. Economic factors have also led to termination of promising programs. One nanoparticle therapy showed modest but real benefits in a rare cancer subtype. The clinical trial was successful, but the market was too small to justify the cost of regulatory approval and commercialization. The company made a business decision to discontinue development and focus resources on more lucrative indications. Patients who might have benefited were left without access, a reminder that scientific success does not guarantee availability. Overestimation of the enhanced permeability and retention effect in humans has been a recurring issue. Many nanoparticle designs rely heavily on passive tumor accumulation, but human tumors often have less leaky vasculature than mouse models. Some patients show minimal EPR effect, and there is currently no reliable way to identify them before treatment. Trials that assume all patients will benefit from passive targeting may dilute efficacy signals when results are averaged across diverse populations. Efforts to develop imaging methods that assess EPR before treatment could help select appropriate patients, but such methods are not yet validated for routine use. Resistance development has thwarted other approaches. Even when nanoparticles successfully deliver drugs to tumors, cancer cells can evolve mechanisms to survive. Efflux pumps that expel drugs, changes in cell signaling pathways, and alterations in cell death machinery all allow tumors to escape nanoparticle-based therapies just as they escape conventional treatments. Combination strategies that attack multiple pathways simultaneously show promise but add complexity and cost. Lessons from these failures have shaped current research priorities. There is greater emphasis on validating targets and mechanisms in human-relevant models before advancing to clinical trials. Researchers are more cautious about extrapolating from mouse data and more attentive to patient selection criteria. Manufacturing process development occurs earlier, with an eye towards scalability and consistency. Immune profiling is incorporated into trial design to identify at-risk individuals. While these measures cannot eliminate failure, they reduce the likelihood of late-stage surprises and improve the overall efficiency of drug development. Failure is an inherent part of innovation. Each unsuccessful program provides data that informs subsequent efforts, gradually refining understanding and improving the probability of future success. The field learns as much from what does not work as from what does. Part 15: Nanoparticles Beyond Cancer. While cancer dominates discussions of nanoparticle therapy, applications extend to other diseases where targeted delivery or controlled release offer advantages. Infectious diseases, cardiovascular conditions, autoimmune disorders, and neurological diseases all present opportunities for nanoparticle-based interventions. Infectious diseases, particularly drug-resistant bacterial and fungal infections, are targets for nanoparticle therapy. Antibiotics delivered via nanoparticles can achieve higher concentrations at infection sites, potentially overcoming resistance or reducing the doses needed to clear pathogens. Liposomal formulations of amphotericin B, mentioned earlier, exemplify this approach. Nanoparticles can also target macrophages, immune cells that harbor intracellular pathogens like tuberculosis bacteria. By delivering antibiotics directly into macrophages, nanoparticles increase drug exposure where it is needed most. Tuberculosis remains a global health challenge, and nanoparticle formulations that reduce treatment duration or improve efficacy could have significant public health impact. Viral infections, including HIV and hepatitis, involve reservoirs of virus in tissues that are difficult to reach with conventional drugs. Nanoparticles that penetrate lymph nodes or cross into the central nervous system could suppress viral replication in these sanctuaries. Antiretroviral drugs delivered via nanoparticles show prolonged release profiles, potentially allowing less frequent dosing and improving adherence. This is particularly valuable in resource-limited settings where daily medication regimens are challenging to maintain. Cardiovascular diseases, including atherosclerosis, benefit from targeted delivery of anti-inflammatory or lipid-modulating agents. Atherosclerotic plaques contain macrophages laden with cholesterol, and these cells can take up nanoparticles. Delivering drugs that stabilize plaques or reduce inflammation could prevent heart attacks and strokes. Several formulations are in preclinical testing, targeting plaque components or vessel walls. The challenge is achieving sufficient drug delivery without causing systemic side effects or disrupting normal vascular function. Autoimmune diseases like rheumatoid arthritis and inflammatory bowel disease involve dysregulated immune responses that damage tissues. Nanoparticles carrying immunosuppressive drugs or anti-inflammatory agents can target inflamed tissues, reducing systemic exposure and side effects. Some formulations exploit the fact that inflamed tissues have increased vascular permeability, similar to tumors, allowing passive accumulation. Others use ligands that bind to markers of inflammation. Early clinical trials show that nanoparticle delivery can reduce disease activity with lower drug doses than conventional formulations require. Neurological diseases present unique challenges due to the blood-brain barrier, which restricts entry of most drugs into the central nervous system. Nanoparticles have been engineered to cross this barrier using various strategies. Some are coated with molecules that bind to receptors on brain endothelial cells, triggering transport across the barrier. Others exploit natural transport pathways or use focused ultrasound to temporarily disrupt the barrier. Potential applications include delivering drugs for Alzheimer's disease, Parkinson's disease, brain tumors, and stroke. Progress is slower than in cancer, partly because neurological diseases are complex and partly because safety concerns are heightened when targeting the brain. Gene therapy for inherited diseases is another frontier. Nanoparticles can deliver corrective genes or gene editing tools to specific tissues, potentially curing genetic disorders. Lipid nanoparticles carrying mRNA have already proven their worth in COVID-19 vaccines, and the same platform is being adapted for rare genetic diseases affecting the liver, muscle, and other organs. CRISPR-based therapies delivered by nanoparticles are in early trials for conditions like sickle cell disease and certain forms of blindness. Vaccine delivery has been transformed by nanoparticle technology. mRNA vaccines for COVID-19 relied on lipid nanoparticles to protect fragile mRNA and deliver it into cells, where it instructed the production of viral proteins that triggered immune responses. This platform is now being adapted for vaccines against influenza, malaria, HIV, and cancer. Nanoparticles can also carry adjuvants, substances that boost immune responses, in controlled release formulations that prolong antigen exposure and enhance immunity. Regenerative medicine and tissue engineering use nanoparticles to deliver growth factors, stem cells, or scaffolding materials that promote healing. After injury or surgery, targeted delivery of factors that stimulate tissue regeneration could accelerate recovery and improve outcomes. This application is largely experimental but represents a departure from traditional pharmacology toward therapies that restore rather than merely treat. Each disease area presents distinct challenges. What works for cancer may not translate to infections or neurological disorders. Nonetheless, the principles of targeted delivery, controlled release, and reduced toxicity apply broadly. As the field matures, nanoparticle therapy will likely find niches across medicine, addressing problems that conventional approaches struggle to solve. Part 16: Patient Advocacy and Community Perspectives. Patients and advocacy groups play roles in shaping nanoparticle therapy development that extend beyond participation in clinical trials. Their voices influence research priorities, trial design, regulatory decisions, and access policies, bringing perspectives that scientists and industry may overlook. Advocacy organizations for specific cancers or diseases often fund research into nanoparticle therapies, directing resources toward areas they perceive as underfunded or promising. These groups collect donations, organize fundraising events, and issue grants to researchers. Their priorities reflect patient concerns, which may emphasize quality of life, reducing treatment burden, or addressing cancers with limited options. By funding early-stage research, advocacy groups help de-risk projects that might not attract commercial investment, enabling exploration of ideas that could eventually prove valuable. Patient input into clinical trial design has increased in recent years. Regulatory agencies and funding bodies now encourage or require patient engagement when planning studies. Patients provide feedback on trial endpoints, helping ensure that what is measured aligns with what matters to those living with disease. Survival is important, but so are symptom control, treatment convenience, and side effects. Patients may prioritize different outcomes than researchers, and incorporating their perspectives can make trials more relevant and acceptable. Recruitment and retention in clinical trials depend on patient willingness to participate. Advocacy groups help by raising awareness of trials, explaining what participation involves, and connecting interested individuals with trial sites. They address misconceptions, provide support for those considering enrollment, and advocate for trial designs that minimize burden. Travel requirements, frequency of visits, and invasive procedures can deter participation, and patient advocates push for more accessible trial structures. access to investigational therapies outside of trials, through mechanisms like expanded access or compassionate use, is an area where patient advocacy is particularly active. When a nanoparticle therapy shows promise in trials but is not yet approved, patients with no other options may seek access. Advocacy groups help navigate regulatory processes, apply pressure on companies and agencies to grant access, and provide information to patients and physicians. These efforts sometimes succeed, though companies are not obligated to provide investigational drugs and often cite capacity constraints or safety concerns. Post-approval, advocacy groups monitor whether therapies reach patients as intended. They track insurance coverage decisions, highlight disparities in access, and lobby for policy changes that expand availability. When nanoparticle formulations are priced beyond what many can afford, advocates push for price reductions, assistance programs, or government intervention. Their campaigns bring public attention to access issues and create pressure for action. Online communities and social media have amplified patient voices. Forums where individuals share experiences with nanoparticle therapies provide real-world insights into side effects, efficacy, and quality of life that complement clinical trial data. These communities offer emotional support, practical advice, and a sense of solidarity among people facing similar challenges. Researchers and companies increasingly monitor these spaces to understand patient experiences and identify concerns that warrant attention. Patients also contribute to research as partners. Some participate in advisory boards, reviewing research proposals and providing feedback on priorities. Others collaborate in designing patient-reported outcome measures or educational materials. This partnership model recognizes that patients are experts in their own experiences and that their knowledge complements scientific and clinical expertise. Mistrust and skepticism exist within patient communities, particularly among groups that have historically been marginalized or exploited in medical research. Concerns about being treated as experimental subjects, about therapies being tested on them before safety is fully established, or about researchers prioritizing publications over patient welfare are real and rooted in history. Building trust requires transparency, respect, and genuine engagement that goes beyond tokenism. The hope that nanoparticle therapy represents can be both empowering and burdensome. Patients may feel pressure to remain optimistic or to keep fighting even when outcomes are poor. Advocacy narratives often emphasize success stories, which can create unrealistic expectations for others. Balancing hope with realism is difficult, and patients benefit from honest discussions about what nanoparticle therapies can and cannot do. End-of-life considerations are part of the patient experience that advocacy sometimes addresses. When nanoparticle therapies fail and disease progresses, discussions shift to palliative care and quality of remaining time. Advocacy groups increasingly support patients through this transition, recognizing that fighting disease is not the only valid response and that care extends beyond curative treatment. Patient advocacy ensures that the development and deployment of nanoparticle therapy remain grounded in the needs and values of those most affected. Their involvement strengthens research, improves trial design, and pushes for equitable access, making the field more responsive and humane. Part 17. Interdisciplinary nature of the field. Nanoparticle therapy exists at the intersection of multiple disciplines, and progress depends on collaboration among scientists and engineers with diverse expertise. No single field possesses all the knowledge needed to design, test, and deploy these complex systems, making interdisciplinary work essential. Materials scientists contribute fundamental knowledge about how substances behave at the nanoscale. They synthesize new polymers, lipids, and metallic compounds with properties tailored for biological applications. Understanding how surface chemistry affects protein binding, how particle shape influences cellular uptake, and how composition determines degradation rates requires deep knowledge of physical chemistry and materials engineering. These specialists develop the building blocks from which nanoparticle therapies are constructed. Biomedical engineers bridge the gap between materials and medicine. They design nanoparticle systems with specific functions, testing how particles interact with cells and tissues. Engineering principles guide decisions about size, charge, and cargo loading. Biomedical engineers also develop devices and methods for characterizing nanoparticles, ensuring that what is synthesized matches design specifications. Their work translates abstract concepts into tangible prototypes that can be evaluated in biological systems. Biologists provide insight into disease mechanisms and cellular processes. Understanding how cancer cells differ from normal cells, how immune systems recognize foreign materials, and how barriers like the blood-brain barrier function informs nanoparticle design. Biologists conduct experiments to determine whether particles reach their targets, whether cargo is released appropriately, and whether intended biological effects occur. Their expertise ensures that engineered systems align with biological reality. Pharmacologists study how drugs move through the body, how they are metabolized, and how they exert therapeutic effects. For nanoparticle therapies, pharmacokinetics becomes more complex because both the particle and its cargo must be tracked. Pharmacologists measure drug concentrations in blood and tissues, model distribution patterns, and predict optimal dosing regimens. They also assess toxicity, identifying safe dose ranges and potential interactions with other medications. Immunologists contribute understanding of how the immune system responds to nanoparticles. Complement activation, antibody production, and macrophage clearance all fall within their domain. Immunologists help design particles that evade immune detection or, when desired, stimulate immune responses. Their input is critical for predicting and managing immune-mediated toxicities and for developing nanoparticle-based vaccines and immunotherapies. Oncologists and other physicians provide clinical perspective. They identify unmet needs, suggest which patients might benefit from nanoparticle therapies, and design trials that answer clinically relevant questions. Physicians also contribute real-world data on how treatments perform outside the controlled environment of trials. Their involvement ensures that research addresses practical problems and that findings translate into better patient care. Regulatory scientists navigate the pathways to approval. They interpret agency guidance, design studies that meet regulatory requirements, and compile documentation for submissions. Regulatory expertise is essential for moving products from laboratory to market, and specialists in this area bridge scientific innovation and legal compliance. Ethicists examine the implications of nanoparticle therapy, raising questions about consent, equity, and responsible innovation. Their work ensures that ethical considerations are integrated into research planning and that potential harms are anticipated and addressed. Patient advocates, as discussed earlier, bring the voices of those affected by disease into research conversations. Their participation ensures that scientific priorities align with patient needs. Data scientists and computational modelers increasingly contribute to nanoparticle design. Machine learning algorithms analyze large data sets to identify patterns and predict which particle characteristics will perform best. Computational models simulate how particles behave in the body, reducing the need for extensive animal testing and accelerating the identification of promising candidates. Manufacturing specialists scale laboratory syntheses to commercial production. Their expertise in process engineering, quality control, and regulatory compliance transforms research findings into reproducible products. Without this expertise, even the most promising nanoparticle would remain a laboratory curiosity. Collaboration across these disciplines is not always seamless. Different fields use different terminologies, prioritize different outcomes, and operate on different timelines. Materials scientists may focus on elegant synthesis methods, while clinicians care only whether the product works in patients. Bridging these perspectives requires communication, mutual respect, and willingness to learn from other disciplines. Funding structures sometimes hinder interdisciplinary work. Grants are often organized by discipline, and proposals that cross boundaries may not fit neatly into review panels. Institutions are structured into departments that can create silos, making collaboration logistically challenging. Efforts to promote interdisciplinary research, such as dedicated centers or training programs, help overcome these barriers but require sustained commitment. The most successful nanoparticle therapies emerge from teams where diverse expertise is integrated from the beginning. Early involvement of clinicians prevents designs that are scientifically interesting but clinically impractical. Early involvement of regulatory specialists avoids pathways that cannot lead to approval. Early involvement of patients ensures that the therapy addresses real needs. Interdisciplinary collaboration is not merely desirable but essential for translating scientific potential into therapeutic reality. Part 18. Measuring Success and Defining Progress. Evaluating progress in nanoparticle therapy requires clear metrics, but defining success is not straightforward. Different stakeholders prioritize different outcomes, and what constitutes meaningful progress depends on perspective and context. For researchers, success may be measured by publications, citations, and grants. Discovering a novel nanoparticle mechanism or demonstrating proof of concept in a preclinical model advances scientific knowledge and careers. These metrics drive academic incentives but do not directly reflect clinical impact. A highly cited paper may describe a system that never reaches patients, While a clinically important advance may go unnoticed in academic circles if it involves incremental engineering rather than conceptual novelty. For companies, success is defined by regulatory approvals, market share, and profitability. A nanoparticle therapy that gains approval and achieves commercial viability is a success regardless of whether it represents a major scientific advance. Conversely, a scientifically elegant system that fails in trials or cannot be manufactured economically is a failure from a business perspective. These incentives shape which projects receive investment and which are abandoned. For regulators, success means ensuring that approved products are safe and effective. Metrics include the quality of trial data, the robustness of manufacturing processes, and the adequacy of post-market surveillance. Regulators balance the need to protect patients with the desire to facilitate access to beneficial therapies, a tension that varies by disease severity and available alternatives. For patients, success is improvement in survival, quality of life, or both. A therapy that extends life by months but causes severe side effects may or may not be considered successful depending on individual values. Some patients prioritize survival above all else, while others prioritize comfort and dignity. Patient-reported outcomes, including pain, fatigue, and emotional well-being, are increasingly recognized as important measures of success, though they are harder to quantify than survival or tumor response. For healthcare systems, success involves cost-effectiveness and resource allocation. A nanoparticle therapy that improves outcomes but costs substantially more than existing options may strain budgets, limiting access to other services. Systems must balance the benefits to individuals against the broader population's needs, a utilitarian calculus that sometimes conflicts with individual patient interests. Defining progress over time is also complex. Early in the field, simply demonstrating that nanoparticles could deliver drugs to tumors was a significant achievement. As the technology matured, the bar rose. Now, incremental improvements in targeting efficiency or toxicity reduction, while valuable, may not generate the same excitement. The field risks being judged against unrealistic expectations of revolutionary breakthroughs when steady, incremental progress is the more likely trajectory. Benchmarking against conventional therapies provides one measure of progress. If a nanoparticle formulation offers no advantage over existing treatments, it adds little value despite technical sophistication. However, even modest improvements can be meaningful. Reducing cardiac toxicity in chemotherapy, extending progression-free survival by weeks, or enabling treatment in patients who previously could not tolerate it all represent real progress, even if they do not constitute cures. Surrogate endpoints in clinical trials, such as tumor shrinkage or biomarker changes, are used to assess efficacy more quickly than waiting for survival data. However, surrogates do not always predict survival benefit, and therapies that look promising by surrogate measures sometimes fail to extend life. Balancing the need for rapid assessment with the imperative to demonstrate true clinical benefit is an ongoing challenge. Long-term outcomes, including durability of response and late effects, are essential for understanding the full impact of nanoparticle therapies. Trials typically follow patients for months to a few years, but effects that emerge over decades may not be captured. Registries and post-market studies provide longer follow-up, but participation is often incomplete and data quality varies. The concept of personalized or precision medicine complicates success metrics. If a therapy works well in a subset of patients but not others, is it a success? Identifying biomarkers that predict response could allow selective use, maximizing benefit and minimizing harm. However, such stratification reduces the potential market, which may deter investment. The tension between broad applicability and targeted efficacy shapes development priorities. Negative results, trials that do not show benefit, are also a form of progress. They eliminate approaches that do not work and inform future efforts. However, negative results are less likely to be published, a bias that distorts the literature and leads to wasted effort as others repeat failed experiments. Encouraging publication and dissemination of negative findings would improve the field's efficiency. Measuring success in nanoparticle therapy ultimately requires balancing scientific rigor, clinical relevance, economic reality, and patient values. No single metric captures all dimensions, and progress is best understood through multiple lenses. The field advances not in leaps but through accumulated small gains, each building on the last, gradually expanding what is possible. Part 19. Philosophical Reflections on Scale and Complexity. Nanoparticle therapy invites reflection on the nature of medical intervention and humanity's relationship with technology. The act of engineering particles at the molecular scale to navigate the body's intricate systems embodies both profound capability and humility before biological complexity. Scale itself is philosophically significant. The nanometer range is beyond direct human perception, accessible only through instruments. We design systems we cannot see, relying on models and indirect measurements. This requires trust in scientific methods and acceptance of abstraction. The particles we engineer operate in a world governed by forces different from those experienced at human scale. Surface tension, Brownian motion, and quantum effects shape nanoparticle behavior in ways that defy everyday intuition. Working at this scale demands thinking that bridges the tangible and the theoretical. Complexity in biological systems humbles even the most sophisticated engineering. The body is not a simple machine but an adaptive, self-regulating network of cells, tissues, and organs. Introducing nanoparticles perturbs this system in ways that are difficult to predict fully. Interactions cascade through multiple levels of organization, from molecular binding to cellular responses to immune activation to organ function. Designing a nanoparticle that performs as intended requires understanding these layers, yet our knowledge remains incomplete. Every intervention is an experiment, a dialogue with biology whose outcomes are uncertain. The protein corona exemplifies this complexity. Within moments of entering the bloodstream, a nanoparticle is no longer the designed object but a hybrid entity coded with proteins. The particle's identity is fluid, determined as much by the biological environment as by engineering choices. This challenges the notion of control and highlights the limits of reductionist approaches. We cannot simply design a particle in isolation. We must design for interaction with a dynamic, context-dependent system. Targeting raises questions about specificity and collateral effects. No targeting strategy is perfectly selective. Even the best systems deliver some drug to unintended sites, and every intervention affects more than its primary target. The goal is not absolute precision but acceptable trade-offs. This pragmatic approach contrasts with idealized visions of molecular medicine that promise surgical strikes against disease with no side effects. Reality is messy, and acknowledging this honestly is essential. The concept of natural versus artificial blurs in nanoparticle therapy. Lipid nanoparticles mimic cell membranes, using the body's own materials. Biomimetic coatings disguise synthetic cores as biological entities. Are these particles foreign or familiar? The question matters for immune recognition and ethical considerations about what belongs in the body. The boundaries between organism and technology become porous, challenging dualistic thinking about biology and engineering as separate realms. Human agency in medicine reaches new dimensions with nanoparticle therapy. We no longer merely administer substances and observe effects. We engineer systems that act autonomously within the body, making decisions at the cellular level about when and where to release drugs. This delegation of agency to designed objects raises questions about responsibility and oversight. If a particle malfunctions, causing harm, where does accountability lie? These are not merely legal questions but philosophical ones about the nature of action and intention. Hope and hype are constant companions in emerging medical technologies. Nanoparticle therapy has been described in terms that suggest revolutionary transformation, yet progress has been incremental. Managing expectations is critical to maintaining public trust and ensuring that disappointment does not overshadow genuine achievements. The tension between inspiring hope, which motivates patients and attracts funding, and maintaining realism, which prevents overreach, is difficult to navigate. The long arc of scientific progress provides perspective. Medicine has always involved tools that extend human capability, from scalpels to antibiotics to imaging devices. Nanoparticles are the latest addition, remarkable for their scale and sophistication but continuous with a tradition of using technology to address suffering. Placing nanoparticle therapy within this broader history tempers both excessive enthusiasm and unwarranted pessimism. Reflecting on nanoparticle therapy also means confronting mortality and limits. These technologies may extend life or improve its quality, but they do not eliminate the fundamental vulnerability of human existence. Diseases evolve, bodies age, and not all problems have technological solutions. Medicine, including nanoparticle therapy, operates within constraints imposed by biology, economics, and ethics. Recognizing these limits does not diminish the value of the work but situates it within a realistic understanding of what is possible. The beauty in nanoparticle therapy lies not just in its technical achievements but in what it represents: human ingenuity applied to alleviating suffering, the willingness to venture into the unknown, and the persistence to refine approaches through repeated failures and modest successes. It is work conducted at the edge of perception and understanding, requiring both precision and humility. Whether it fulfills its promise remains to be seen, but the effort itself reflects something fundamental about the human drive to heal and to improve the conditions of life. Part 20. Synthesis and Looking Forward. Nanoparticle therapy represents decades of accumulated knowledge, incremental progress, and interdisciplinary collaboration. From early observations that particles accumulate in tumors to sophisticated systems that respond to biological signals, the field has evolved through careful experimentation and learning from both successes and failures. The approved formulations in clinical use, while not revolutionary, provide real benefits to patients by reducing toxicity and improving drug delivery. Research continues on more advanced systems that integrate targeting, stimuli-responsiveness, and combination therapies. The challenges that remain are substantial. Most nanoparticles still fail to reach tumors in high concentrations. The immune system clears particles efficiently, and tumor heterogeneity limits efficacy. Manufacturing at scale is expensive and technically demanding. Predicting which patients will respond remains difficult, and biomarkers for patient selection are largely lacking. Safety concerns, though manageable for approved products, require ongoing vigilance as new formulations are developed. Regulatory pathways are rigorous, appropriately so, but add time and cost to development. Looking forward, the field will likely continue on its current trajectory of gradual improvement rather than sudden breakthroughs. Advances in materials science will yield particles with better biocompatibility and more sophisticated release mechanisms. Computational modeling and artificial intelligence may accelerate design and reduce dependence on trial and error. Integration with immunotherapy, gene therapy, and other emerging modalities will expand applications beyond traditional chemotherapy. Personalized approaches that tailor nanoparticles to individual tumor characteristics may eventually become feasible, though significant technical and economic barriers must be overcome first. Applications beyond cancer, in infectious diseases, autoimmune conditions, cardiovascular disorders, and neurological diseases will grow as the principles of targeted delivery and controlled release are adapted to these contexts. Each disease area presents unique challenges, but the fundamental advantages of nanoparticle systems apply broadly. Vaccines, gene therapies, and regenerative medicine will increasingly rely on nanoparticle delivery platforms. Global disparities in access remain a pressing concern. Most innovation occurs in wealthy countries, and most approved therapies are available primarily to patients in resource-rich settings. Addressing this inequity requires concerted effort from governments, industry, international organizations, and advocacy groups. Generic formulations, technology transfer, and tiered pricing could expand access, but political will and sustained commitment are essential. Patient voices will continue to shape the field. As advocacy grows more sophisticated and patient engagement becomes routine in research and regulatory processes, priorities will shift toward outcomes that matter most to those living with disease. Quality of life, convenience, and reduced burden of treatment will receive greater emphasis alongside survival and tumor response. Ethical considerations will evolve as technologies become more powerful. Questions about consent, equity, dual-use risks, and environmental impacts will require ongoing attention. The field must balance innovation with responsibility, ensuring that progress serves the common good and does not exacerbate existing inequalities or create new harms. Interdisciplinary collaboration will remain essential. No single discipline can address the complexities of designing, testing, and deploying nanoparticle therapies. Materials scientists, biologists, engineers, clinicians, ethicists, patients, and many others must work together, bridging differences in language, priorities, and methods. Institutions and funding agencies that support such collaboration enable progress that would be impossible within disciplinary silos. Measuring success requires recognizing that progress is incremental and multifaceted. Small improvements in targeting, modest reductions in toxicity, and incremental increases in survival all represent meaningful advances. The field should be judged not against idealized visions of perfect cures, but against the realistic standard of whether patients are better off than they were before. By this measure, nanoparticle therapy has already contributed value and holds promise for continued contributions. The work is far from complete. Many diseases remain intractable, and even the best nanoparticle systems have limitations. Resistance develops, side effects occur, and not all patients respond. The path forward involves persistence, learning from setbacks, and maintaining realistic expectations while pursuing ambitious goals. Science advances through accumulated effort, each study adding to collective knowledge, each failure refining understanding, each success opening new possibilities. Nanoparticle therapy exists at a unique intersection of scale, complexity, and human need. It operates in a realm beyond direct perception, guided by theory and measurement, striving to manipulate biological systems in ways that alleviate suffering. The particles themselves are silent actors, traveling through blood and tissue, interacting with cells according to the properties engineered into them. Whether they deliver their cargo successfully, whether tumors shrink, whether lives are extended or improved, depends on countless variables and interactions that cannot be fully controlled or predicted. This uncertainty does not diminish the value of the work, but is intrinsic to it. Medicine has always involved navigating uncertainty, making decisions with incomplete information, and accepting that outcomes vary. Nanoparticle therapy amplifies both the possibilities and the complexities, offering new tools while demanding deeper understanding of the systems those tools engage. The future will bring new materials, better targeting strategies, more sophisticated release mechanisms, and integration with technologies not yet imagined. It will also bring disappointments, failures, and the realization that some problems are harder than anticipated. Both are part of the process. The field will advance through the efforts of thousands of researchers, clinicians, patients, and others who contribute in different ways toward the shared goal of better treatments. Nanoparticle therapy is neither miracle nor mirage, but a practical approach to solving specific problems in drug delivery and therapeutic efficacy. Its trajectory over coming decades will depend on continued investment, rigorous science, ethical vigilance, and commitment to ensuring that benefits reach those who need them. The particles themselves, invisible and engineered with exquisite care, carry more than drugs. They carry hope, effort, and the accumulated knowledge of a field still learning, still striving, still pushing at the boundaries of what medicine can accomplish. In the quiet hours, when reflection comes easily, it is worth considering that these microscopic systems represent something larger. They embody human creativity applied to fundamental challenges of health and disease. They illustrate the power and limits of technology. They remind us that progress is rarely linear or complete but is nonetheless real and valuable. And they offer a glimpse of a future where medicine operates with greater precision, less harm, and more attention to individual needs, even as they remind us that such a future requires patience, humility, and sustained collective effort. The story of nanoparticle therapy continues.